was the first farmer who planted a seed and grew our world? It was the birth of progress. Because someone could grow a little corn, some wheat, rice, humanity could grow everything else. This is the power of farming. The power to grow our civilization. But who could have predicted how big civilization could get? Our population is increasing. Our natural resources are declining, and our planet's climate is changing. So farming needs to change too. And it has, with smarter tools, sustainable practices, and ever higher yields. Farming gives progress to the world, and the world is giving it back to farming. We're Corteva Agriscience, and we're here to support the people who support us all. With stronger seeds, precise crop protection, and data you can count on, we're bringing the next generation of farming to the next generation of farmers.
Sustainability. It's something we all aspire to. But like any ambition, sustainability will only happen if we make it happen. At Corteva Agriscience, 21,000 employees around the world are working every day to do just that. Last year, we set 14 sustainability goals to hit by 2030, and we're on our way to meeting them. We're in it for farmers. We're helping farmers boost yields around the world and are working with them to make their livelihoods as resilient as they are. We're in it for the land. We're helping farmers and ranchers make strategic land management decisions, improving the soil, conserving water, and enhancing biodiversity. We're in it for our communities. From accelerating COVID-19 testing in Iowa to distributing food in Latin America, our response to the pandemic showed our commitment to helping protect people throughout the food system. We're in it for the planet. We're committed to reducing greenhouse gas emissions and becoming a more carbon-efficient business throughout our operations. Across Corteva, sustainability isn't just part of our business, it is our business. We're all in it together, and we're in it for good. Download our first sustainability report to see how.
morning, everyone. We will start in 1 minute. Karen, we'll start in 1 minute. Hey.
Okay, good morning, everyone, especially those attending via Zoom and those who are watching via YouTube Live. This week, we are celebrating the 19th National Biotechnology Week with the theme Empowering Innovation for Sustainable Future of Biotechnology. This year's celebration is led by the Department of Agriculture through the DA Biotech Program Office. So today's webinar is the second installment of the series titled Biotech Innovations for a Sustainable Agriculture. This webinar was organized by the International Service for the Acquisition of Agri-Biotech Applications, Incorporated, in partnership with the DA Biotech Program Office, Bureau of Animal Industry, Biotechnology Coalition of the Philippines, and Corteva Agriscience. I am Ms. Karen Penasa, and I will serve as your facilitator and moderator for today. So before we proceed, let me acknowledge our partners from ISA Incorporated. We have Executive Director Rodora Aldamita. Good morning, ma'am.
Morning.
We have also the Director of the DA Biotech Program, Dr. Claro N. Mingala. Good morning, sir.
Morning to everyone.
We have also from Corteva Agriscience, Dr. Antonio Alfonso. Good morning, sir.
Mute.
Oh, yes. Good morning. Good morning, everyone.
All right. So before we proceed with the program, allow me to quickly remind you of the house rules. So for proper flow of the program, Zoom attendees are requested to keep their microphones on mute unless invited to speak. We highly encourage our audience to engage and actively participate during the open forum. All questions will be addressed during the open forum. The online audience is requested to post your questions using the Zoom chat box or via the YouTube Live. Also, please accomplish the online evaluation form to receive your certificate via email after the event. The link and the QR code to the evaluation form will be flashed at the end of the webinar series. Okay, so biotechnology has the potential to revolutionize agriculture and make it more sustainable. This webinar series explores the role of biotech innovations in driving sustainability, providing solutions to some of the most pressing problems in the agriculture sector, as well as the various challenges surrounding it. This webinar intends to highlight the recent advancements and initiatives on GM animal development globally and the significance of a robust regulatory framework for sustainable GM animal development. It also aims to highlight biotechnological approaches to meet the demand for seafood through aquaculture, as well as to discuss the recent advancements in food biotechnology and tackle eco-friendly bio-innovations as an alternative to plastic. So without further ado, may I now call on Mr. Elijah M. Avante to deliver the opening message on behalf of the Bureau of Animal Industry Assistant Director, Arlene Asteria V. Vichaco.
So good
Sir
morning,
Elijah, please take it
everyone.
away.
Yes, thank you, Karen. So good morning, everyone. Allow me to deliver the welcome remarks for the webinar on biotech innovations for sustainable agriculture. So our distinguished partners, guest speakers, policymakers, members of the academe, and all the stakeholders from various parts of the country who are joining us virtually, we extend our sincere appreciation and warm greetings to all of you. We are honored and delighted to welcome everyone to the second installment of the webinar series titled Biotech Innovations for a Sustainable Agriculture. So co-organized by our colleagues from the DA Biotechnology Office, the International Service for the Acquisition of Agri-Biotech Applications, the Biotechnology Coalition of the Philippines, Cordeva AgriScience, and us from the Bureau of Animal Industry. So this webinar series is a part of the build-up activities in celebration of the 19th National Biotech Week. So the extremity on multifariousness of the issues and challenges faced by the agriculture sector underscore the need for a scrupulously designed solution. So biotech innovations hold the potential in revolutionizing interventions with greater precision. So the Sustainability Development Goals adopted by the United Nations in 2015 promote integrated and multidisciplinary approaches to help address the pressing issues affecting our world today. So ultimately, these goals aim to foster developments and balance social, economic, and environmental aspects, recognizing their interplay in bolstering a sustainable future. So today's webinar will delve into developments in animal biotechnology research, the application of biotech approaches in aquaculture, and importance of robust policy frameworks to facilitate the safe use and adoption of modern biotechnology. Over the course of this webinar, we will have the privilege of learning from some of our notable experts in the field, namely Dr. Marvin A. Villanueva, Dr. Casiano H. Coresca Jr., and Dr. Claro N. Mingala, all confirmed as a career scientist through the scientific career system. We encourage you to actively participate, ask questions, and engage in discussions with our speakers during the open forum. It is through this exchange of knowledge that we can collectively harness the potential of innovations. So in closing, we would like to extend our heartfelt gratitude to our partners, speakers, and most importantly, to each and every one of you who has joined us today. Your participation is a testament to our commitment in creating a sustainable, food secure, and resilient Philippines. We hope that by the end of this webinar series, you will leave with a deeper understanding of the transformative power and role of innovations in driving sustainability and an even greater sense of optimism about the future of agriculture. So thank you, and may all of you, all of us, enjoy the insightful discussion ahead. So on behalf of our Director Paul Limson, and our Chairperson, Assistant Director Arlene Asteria Vichaco. Pleasant morning to everyone, and I am once again Eliza Emma Vante from the Head Secretariat and Vocal Representative of the BAI Biotechnology.
Thank you very much, Sir Elijah, and thank you, of course, to our BAI Director Paul Limson, and BAI Assistant Director Dr. Arlene Asteria Vichaco for the inspiring message. Indeed, sustainable agriculture driven by biotechnology is not only a possibility, but a necessity for our future. So I'm certain that our 290 attendees cannot wait to listen to today's presentations. So let me now introduce our first speaker to talk about genetically modified animals, developments in research and policy framework. He finished his bachelor's degree in animal husbandry and Doctor of Veterinary Medicine degree at Central Luzon State University. He acquired his PhD in veterinary medicine at Hokkaido University in Japan. He has more than 10 years of experience in R&D, and his current research include food safety, particularly for antimicrobial resistance, adulterants, and veterinary drug residues, including antibiotics, growth promotants, and other contaminants in food, livestock biotechnologies, epidemiology, detection, and control of bacterial zoonotic diseases, addressing operational health problems in ruminant farming in the Philippines. He is currently the chief of the DA Livestock Biotechnology Center and officer in charge, chief of the Research and Development Division of the Philippine Carabao Center, National Headquarters, and Gene Pool. He is also a career scientist. Ladies and gentlemen, join me in welcoming Dr. Marvin A. Villanueva. Good morning, sir.
Good morning. Thank you very much, Miss Yen, for the introduction. So let me first share my slide. Okay, can you see it now, Miss Yen?
Yes, sir.
Okay. So again, good day, everyone, and thank you very much for the organizers for inviting me to participate in this webinar series on biotech innovations for a sustainable agriculture. Um, so since some of you already attended the first day of this webinar series, which is focusing on plant biotechnology this time, we will be focusing on animal biotechnology. And this is a build-up activity for this upcoming 19th National Biotech Week with the theme Empowering Innovation for Sustainable Future with Biotechnology. So in this topic, I will be focusing more on the recent developments in research on GM or gene-edited animals, and I will provide a very brief overview on the policy framework on GM animals in the Philippines, since there is a detailed presentation later in this webinar today. Okay, just a moment. Just for a moment. Okay. Okay. Okay, so the world population continues to increase, and feeding the world sustainably remains to be a global challenge. The forecast in this chart as shown that the world population will exceed by 10 billion by 2059, while the annual decline in population growth is also presented in this forecast. There is an urgent need to find innovative ways to meet this challenge, taking into consideration preserving the natural ecosystem for future generations. Land resources are considerably finite resources, and pushing for land conversion for agricultural expansion can lead to biodiversity loss, soil degradation, and an increase in greenhouse gas emissions. The main challenge now is how do we produce more with less? An increase in population also translates in the widest variety of preferences and a change in consumer patterns. And in this aspect also adds pressure to the produce specific agricultural products to meet this demand. Strategies must be employed to curb this increase in demand. So agriculture, as we all know, is a significant contributor of greenhouse gases emissions. Livestock production contributes to about 11 to 17% of global GHGs emissions of nitrous oxide from fertilizer use also increased over the last 10 years, as well as carbon dioxide emission from land use changes in energy consumption. Another problem is climate change that significantly impacts on food system worldwide, affecting food production, distribution, and access to nutritious food. These impacts pose considerable challenges to global food security and can exacerbate vulnerabilities in developed and developing regions in the world. So in the same way, meat from livestock is an important source of nutrition for many people around the world. Global demand for meat is growing with over the past 50 years. Meat production has more than tripled. The world now produces more than 340 million tons each year. So the production of meat has large environmental impacts also, increasing greenhouse gas emissions, agricultural land, and freshwater use. So one of the world's most pressing challenges is to produce and consume meat, dairy, and other protein products in the way that reduces its environmental impacts. In addition, the livestock industry poses severe and constant challenges. Among these are the adverse environmental conditions such as climate change. Another problem is the lack of new breeding stocks, since some other livestock animals do not have a developed breeding lines locally and usually depend on imports. There's also the problem on high cost of farm inputs, as exemplified by the high prices of feeds among non-ruminants and ruminants, since we are also highly dependent on imports. And also the constant threat of the transboundary diseases such as African swine fever, avian influenza, lumpy skin disease, and the like. Okay, so the reason why we think biotechnology is one of the important tools that we can use is this can be a game changer to address this problem. So first, let us discuss first what is biotechnology. So biotechnology is the use of biology to develop new products, methods, and organisms intended to improve human health and society. So biotechnology in livestock or animals can greatly improve the animal productivity in various ways, namely by improving the production of products, by promoting growth and improving nutrient intake efficiently, by increasing the rate of reproduction of domestic animals, increasing the quality of animal production, resistance to diseases, and many more. So for the animal or livestock biotechnology, it has been used for a long time actually, as evidenced by these pictures on the left, beginning as far as 8,000 years ago. So usually what they do is the traditional animal biotechnology. And as you can see on the right side, I think some of you are familiar with this traditional animal biotechnology, such as improvement in the production of chickens. So from before, in order to have a 1 kilogram chicken for 56 days, right now we can harvest a 1 kilo chicken for only 21 days. So that is a very very big improvement in terms of the production to feed the number of people worldwide. Another is selective breeding to come up with a desired trait of a certain livestock. Use of artificial insemination to maximize the use of this good genetic trait from selected bulls. Use of vaccines to prevent diseases, and so on. So it's only when the discovery of the genetic code or the sequencing of the genomes of different livestock animals started the use of the modern animal biotechnology. Okay, so here are some of the applications or benefits of the livestock biotechnology as a tool. So it can be used for climate change and disease resilient or development of resistant animals, increased income for livestock food producers, doubling food production to meet the supply and demand, and proper animal management, and lastly, to develop rapid diagnosis and conduct modern disease surveillance. Okay, so for the introduction of this GM animals as defined involves the altering of its genetic material by adding, changing, or removing certain DNA sequences in a way that it does not occur naturally. It aims to modify specific characteristics of an animal or introduce a new trait such as disease resistance or enhanced growth. So for the side of animal, we have two methods to develop GM animal. One is through transgenesis or cisgenesis, which is the transfer of genes in an animal. And another one is through the deletion of genetic of a gene or genetic information. So aside from the one that I mentioned a while ago, through the developments in biotechnology, researchers were able to hasten or make it faster using this biotechnological tools. They come up with this gene editing technology. In the simpler explanation, it is like it works as like a biological version of a word processing program's find and replace functions. For example, we have this CRISPR-Cas system wherein there is a guide molecule and a DNA cutting enzyme, and through this technology, we can remove those defective DNA strand and replace it with a healthy strand that we can use nowadays. So genome editing or gene editing is the precise targeted alteration of the DNA sequence in a living cell. So in farm animal breeding, it could be used to produce animals with specific traits that might be difficult to achieve using conventional selective breeding approaches. So in a nutshell, these are just some of the new breeding innovation as a tool that we can use as a driver of change in livestock. So usually we use the cell source such as zygote and fibroblast, and through this gene editing technology such as CRISPR-Cas system, we can knock in or knock out, and also we can do point mutation on a specific target gene, do cell manipulation such as inoculation, nuclear transfer, cloning. Sometimes we use the somatic cell nuclear transfer, and then do the embryo transfer in different livestock animals, and we can do it in various applications such as in agricultural and biomedical applications. Okay, so let me just show you this very good figure from the paper of Van Ininam et al. In 2021, which is entitled Genetic Engineering of Livestock, the Opportunity Cost of Regulatory Delays, wherein actually the start of this modern animal biotechnology started in 1985, where the first transgenic pig and sheep was developed. And up to now, it's about 38 years since the first introduction of this transgenic pig and sheep. So as you can see in this figure, these are some of the milestones in producing the GE livestock. So unfortunately, as compared with the crops using advanced biotechnology, very few of these milestones were commercialized for several reasons. So maybe later it will be discussed in other presenters. But so far around the world there are many um, um, many innovations that were developed by different researchers around the world. So in the Philippine Carabao Center where I am working, we were able to also develop, or we were able to conduct this somatic cell nuclear transfer, since we have a very robust selective breeding for water buffaloes, and we really want those breeds with the desired traits to be replicated, so that it can help in producing more milk, meat, and other products out of water buffalo. So right now, this somatic cell nuclear transfer product were still under research condition, and it's still within the bounds of the research facility of the Philippine Carabao Center. But it's a proof that the Filipinos can do those advanced livestock biotechnological techniques. So at this moment, let me just introduce you to some of these GE animals that are on the pipeline, that are developed in different parts of the world. Since right now the regulations for GM animals in the Philippines are still in the infancy stage. So it's good that we will be familiar with this products of livestock innovations using biotechnology that we can use later to address those problems in livestock industry. So the first one is the intentional genomic alteration of the slick hair coat to better regulate the internal body temperature with an increased capacity of sweating. So what they did here is to knock out the slick gene, so that this animal on the left side has a thinner or no hair in their skin. So it usually address this current problem on climate change, wherein several animals are severely affected due to a harsh environmental condition. So another, which is developed by the group of Dr. Goats in New Zealand, is the color dilution of dairy cattle, which is lightening of the coat color that can reduce the radiative heat gain from exposure to the sun. So as you can see here on the lower left part of the picture, usually the color of the Holstein Friesian is black and white, but due to knocking out of this premelanosome protein, so you can see here it's from black it became gray. So it's similar with humans. If you wear a dark shirt and you go out during noontime and there is a very sunny weather, you feel very hot. So that this is the reason why these researchers are knocking out this gene to dilute this black color of Holstein Friesian. Okay, so another is the polled cow, as you can see in the upper picture. This is courtesy of Dr. Van Ininam's group. And another one is the gall-safe pigs. So it is a genetically modified animal. It is intended to eliminate the alpha gal sugar on the surface of the pig cells, and those people with alpha gal syndrome may have mild to severe allergic reaction to alpha gal sugar found in red meat such as beef, pork, and lamb. So this technological innovation can be used for both for human for food consumption as well as a source of potential therapeutic uses. Okay, so another is the red Angus. So usually the Angus is a black one, but this group also developed the red Angus also to adapt with the adverse climate nowadays, and also aside from being a producer of good quality meat with good intramuscular marbling, this red Angus also produce excellent milk production and have a strong maternal instinct. Okay, so for the resistant to diseases, this group from Roslin Institute in Europe developed a porcine reproductive respiratory syndrome, or PRRS resistant pigs. So what they do here is it is a gene edited animal that it lacks functional CD163 receptor, which is important in the attachment of this PRRS virus to pigs. Another is they also developed this bird flu resistant chicken, wherein it contains an extra gene that interrupts the transmission of bird flu. So for this bird flu resistant chicken, the researchers focus on the ANP32 that makes the protein of bird flu virus hijacks to replicate itself. What it does, it disrupts the interaction and prevents the protein from being used. Thereby, the virus would not be able to replicate. So let me remind you that all of these products of research are still in the bounds of their research facilities. Okay, so another is what they call Rosita Isa, which was born a cattle born that expressed milk containing proteins present from human milk but lacking in cow milk. So it is a GM animal also, and the goal of these researchers was to raise the nutritional value of cow's milk by adding the two human genes, the lactoferrin, which provide infants antibacterial and viral protection, and lysozyme, which is also an antibacterial agent. And then the last one, although this one is not considered as animal, but the diseases that it carries is important in animals and also in humans. The genetically modified male mosquitoes that carry the self-limiting gene, and the offspring of this mosquitoes do not reach adulthood, since it reduces the spread of mosquito-borne diseases. This is a product from Oxitec. Okay, so aside from this, from the animals that undergone GM or genetic manipulation, there are also other applications of this GM or GE animals. One is the use of GM animal for the to be used as disease models and to improve the treatment strategies in humans, such as this one, the non-human primate, as a model for AIDS and also for polio. So as compared to those GE animals for agriculture or for food, these animals that is used for biomedical research is more advancing than the previous one. So work is still in progress in developing new models by altering the susceptibility of mice to pathogens of humans. Okay, so another is the use of these GM animals for gene therapy. As example for this is the cystic fibrosis. And then the last one is the for xenotransplantation. So I think some of you are aware of some of the organs of animals, such as pigs, can be used as a alternative organs for humans. Like for example, the heart or the kidneys from these GM animals for biomedical purposes. Okay, so let me move on to the GM animal regulatory policy. So the name of this GM animal regulatory policy in the Philippines is the also JDC circular series 1 number 1 series of 2022. It is different from the revised JDC in crops, which is a series of 2021, wherein the title is the rules and regulations for research and development, handling and use, transboundary movement, release into the environment, and management of genetically modified animal and animal products derived from the use of modern biotechnology. So this year, this draft JDC was subjected to several stakeholder consultations around the country. But as of this moment, this regulatory policy is still under review of the Department of Agriculture. So later, I think the Dr. Mingala will provide a more detailed updates on this JDC on GM animals. For the applicability of this JDC, it is used for genetically modified fisheries and other aquatic resources, domesticated animals and biological products used for animal husbandry and veterinary purposes, and lastly, biological agents used for biocontrol derived from the use of modern biotechnology and contain novel combinations of genetic materials. So please take note that the products of gene editing that do not contain novel combinations of genetic materials are not covered in this draft circular. Okay, so as a way forward, the government acknowledges the importance of cooperation in accelerating research and innovation, addressing common challenges, regulatory harmonization, knowledge exchange and capacity, shared expertise, and enhance trade and economic growth. So as a way forward, the following strategies will be employed, can be employed to ensure an enabling environment for biotechnology undertakings in the Philippines. So first is the implementation of clear, predictable, science-based and risk proportionate regulations, which I think the groups on NBCP and also in DA Biotech and other stakeholders are doing a great job in developing these regulations. Establishment of an adaptive and responsive policies that can adapt to rapid advancements and emerging technologies. Fostering international cooperations and harmonization of regulatory standards to streamline global biotech developments and facilitate cross-border research and trade. Adequate and sufficient fund for biotech research and innovation. And lastly, we need to do more education and public awareness. So I think that would be the last slide, and thank you very much for your attention, and I will be happy to answer the questions later on in this forum. Thank you.
Thank you very much, Dr. Marvin, for your wonderful presentation. We look forward to everyone's questions. Again, a reminder, please send us your questions via the Zoom chat box. We will get back to them during the open forum. Again, a virtual applause to Dr. Marvin Villanueva. Thank you, sir. All right, moving forward to our next speaker. He finished his bachelor's degree in fisheries, major in aquaculture at Iloilo State University, University of Fisheries, Science and Technology, as a DOST SEI scholar, and graduated cum laude He also completed his master's and doctor of philosophy in veterinary medicine, major in aquatic medicine at Seoul National University, South Korea. His current research includes biotechnological approaches in the production of mudfish, induced breeding of climbing perch using induced spawning hormones, and promoting aquaculture technologies in freshwater fish. He is currently the chief of the DA Fisheries Biotechnology Center, and he is also a career scientist. Our second speaker is no other than Dr. Kashana Churesca Jr. to tackle biotechnology and aquaculture, meeting growing seafood demand sustainably. A virtual round of applause for Dr. Churesca. Good morning, sir.
Good morning, everyone. Thank you, Ms. Yen, for the introduction. Let me share my slide. Once again, good morning, everyone. So this morning, I'm going to present the context on fisheries biotechnology. And it is quite similar to what Dr. Villanueva has been presented on animal biotech. And my presentation is more on the situation here in the Philippines and some of the advanced biotechnologies that are being produced by other countries or other researchers, and our effort here in the Philippines. How can we innovate more for food and for food security, and in terms of the application in fisheries and aquaculture biotechnology. So in the Philippines, we have around a total area of 2 million plus kilometer and 36,000 coastline. And for most of our work rely on fishing, and it's a major sources of livelihood in the country, where there were around 129 million fishers and 135 million fish farmers. This is based on the survey in 2019. And for aquaculture, we ranked 11th for the production in 2019. And as you can see in this picture, we have a lot of species for aquaculture, from tilapia, to our milkfish, shrimps, other indigenous species for our shellfish, and of course, our shrimp and seaweed. And seaweeds is one of the major or the fourth largest producer of aquatic plants here in our country. So this data from the Fisheries Statistical Bulletin in Asia and Philippine Fisheries Profile and FAO United Nations, it is said here that it's around 2.19 million fisher folks engaged in fishing activities. And it's good now that the Bureau of Fisheries have already registered fisher folks in order for us to monitor our fisher folks and our fish farmers. And around 1.89 billion aquaculture production, including fish, crustaceans, and mollusks. So there are a lot of diverse aquatic animals and plants being produced for aquaculture. And it is said that around 40 kilograms per year, the average capita per Filipino fish consumptions. And this data shows some of the production from 2015 to 2020. So as you can see here, there are some decline in total production for fisheries, and this is categorized either for capture, marine, inland, and aquaculture. And it is said that aquaculture, it is being practiced, yung sabi ko kanina, that I said that it's for freshwater, brackish, and marine water. And there are lots of fish species being produced, and our major production is top 10 in worldwide. And in Southeast Asia, we are ranked 5th. And in the Philippines, our aquaculture is one of the main sources for production. And these three commodities, actually we have 5 top commodities, and there's 3 presented here are tilapia, milkfish, and shrimp. And we have seaweeds, shellfishes, and crustaceans in a row. And according to the production, there's recorded decreasing over the years. And there are a lot of factors being considered for the challenges for our aquaculture in the country. And part of it is the emerging diseases and transboundary movement of aquatic animal diseases. So there are a lot of losses if we could not, mitigate the diseases in our aquaculture farms, and knowingly that if a certain disease attacks our aquaculture stocks, for example, for about 10 or around 20%, no? So most of our stocks will be wiped out. And other factors being considered is the resiliency of the fisheries industry from climate change, calamities, and even economic shocks and pandemics. So last pandemic, so there were already reports that it has been caused during the COVID-19 in our aquaculture industry. And of course, we cannot deny that every year, in some cases in fisheries production, there are decrease or decline of stocks. And I believe there are a lot of factors to be considered for the production. And lately, there are a lot of arguments for the production, from capture to do production in aquaculture in order to boost our fisheries and for food security in terms of aquaculture. So what are those biotech interventions that we could address? So here, I believe that previously Dr. Villanueva said about the definition of biotech. And in fisheries, we categorize it into traditional or modern biotech. And for fisheries, we included here some of the post-harvest, intervention in biotech. Like before, we have our fermentation, like our local patis, our soy sauce, our bagoong, and our tissue culture, especially for seaweeds and biofertilizers. I think there are a big, intervention if we could develop biofertilizers out of the seaweeds, and of course, biocontrol agents. And of course, in fish, we have way before we have already. Some of the other countries develop vaccines, and of course, the use of probiotics. This probiotics has been applied in our feeds, incorporating in our feeds in order to boost nutrition for our fish. And lately, we have our, innovations comes in, and we have our modern biotech. And this modern biotech includes our marker technologies, our omics that has been applied in fish. And we have recently our CRISPR technology. Now, there are other reports, and I will elucidate this later, about some of the produced from CRISPR and our genetic engineering. So if you remember the genetic salmon, that it has been produced by some of the Canadian researchers, that it has like 10 times bigger than the indigenous or the traditional salmon. And here are some of the applications of biotech in fisheries and aquaculture, either in fish breeding, diseases, transplantation. So recently, there were reports that germline stem cells can be also applied in fish. And there are some other countries who are doing this one. So the technique is that there are some fish actually that their life cycle is too long, and they have to adjust they have to transfer germline stem cells to tilapia in order for them to produce, or to reduce the development of the germline cells. And of course, for nutrition biotech, so the use of enzymes and other low-cost inputs in order for us to have a higher protein for our fish, or I mean for feed, used to feed in our fish, and the stem cell technology. So, and of course, other works in genome mapping, characterization, and the like. So those technologies are being applied for improve growth and productivity of our fish, and also to increase resistance to pathogens, and of course to improve the broodstock quality of our fish and produce new products for our fish. And so here are the applications in aquaculture and fisheries. First, for example, for fish breeding. So actually we have our traditional breeding, like for example, in the Philippines, we have already for milkfish, other commodities like tilapia and mud crab and shrimps. And recently, in order for us to fast track, and maybe later on the slides, I'll be focusing on induced breeding in order for us to generate and produce and control reproduction in order for us easily produce fish. And here are some of the products that are being used for induced breeding. So recently, there are commercial products, and with regard to our work in fisheries biotech, so we are optimizing some of the commercial product, and we do have producing our organic hormones in order for us to produce our fish. And then there's a rise of new breeding techniques recently. And as I've said, even before we have our natural selection, we produced many already with these techniques, including our tilapia. So later on, I'll show the photos that we have. We have also artificial selection and our artificial sexual hybridization, the all-male tilapia that we have. And in other reports, there were nuclear transplantation or cloning. However, these are being used, I think before it was in carp, in the goldfish, that's interspecific hybrids, and zebrafish. So mostly for fish are modeled fish. However, for assisted reproduction like tilapia, that they use intracytoplasmic sperm injection. So it's quite, it's difficult and guided reproduction. And should I say, most of this are not yet used for commercial production. Still on the I believe they are optimizing the protocol, but in the context of commercial aquaculture. So recently, it relies only for natural selection or production. And of course, gene transfer. So before we have learned that we have already our GM salmon. So I believe that it was already out in the market in the US, and the company that is being leading this production, I believe it's GA or the Aqua Bounty. So there are recently, this is our for transfixed salmon that we have, and our CRISPR technology. So the sea bream, this was reportedly from Japan, and there are already reports for tilapia in other countries. But however, they are more on the lab scale, I believe, and we are looking for the, some of the publications in order for us to validate these techniques. So And there are other mores for the production of this fish in the lab. So the advantage, of course, for the CRISPR-Cas9 for fish, because the turnover is quite fast, because the reproduction in fish, it takes, for the model fish, it takes a month. And for other big fish, for example, like tilapia, the turnover reproduction is around 4 months. So you can get easily, the production of your breeds, after a year of production. And the other application in the sex control and transgenesis, like for example, the work of Dr. Guerrero in all-male tilapia that has been popularized in the Philippines. And now there are already developing researches, like from the androgen pollens work by CLSU, working on the all-male tilapia. The mechanism or the science behind for that, because if you could have the monosex, like for the all-male tilapia, you can produce the uniform or the the sizes of, you can get a very good harmonized sizes of your cultured tilapia, especially for grow out. And if you could do the all female, so you could have your production, or you can use all females for reproduction. So that's the story behind of having those monosex production of tilapia, for example. And then for aquaculture nutrition, so I think there are already use of biotechnology improved products to use for feeds ingredients, especially if you are having the intensive culture. Especially now we are on the mariculture of different species, and nutrition is necessary to feed and our cultured fishes in cages. So from the natural food production, optimization, those in the laboratory, the isolation of the natural food, and of course, because this is the very core of having hatchery production, and to increase our aquaculture production is the the natural food or the algae that being cultured during the hatchery phase of our culture. And of course, the use of trash fish. So we used to feed fish to fish, and now there are already researchers and other institutions working on the low-cost feeds for our fish, for example, in tilapia and in milkfish. And there are other countries that are developing high proteins also for shrimps and other crustaceans. And this is very important because in developing feeds, we need to be sustainably, for our culture and our production in fish, especially our cultured fish here in the Philippines, because the ingredients or the inputs is very high, and most of us relying our inputs outside. So I think, and I believe we need to have a very intensive researches, especially for our aquatic biotechnology in terms of nutrition, or aquatic, nutrition biotech, in order for us, for example, like developing enzymes and probiotics in order to mitigate diseases also in our cultured fish. Then, of course, for our fish health management, the application is more on, recently is more on detection or early mitigation of diseases. So for fisheries, especially now we are in the Philippines in 2017, it's the first occurrence of the tilapia lake virus, even before the COVID-19. So now our efforts in developing protocol in order for us to mitigate the occurrence of this disease and our bacterial diseases. No? So recently, there are, or in our center, we are developing vaccines and some of the immunostimulants for our fish, especially in tilapia. And for bacterial diseases, so we are also by the use of or application of molecular tools, we are developing protocols in order for us to detect early mitigation. I mean, to detect streptococcosis in deceased tilapia and other fish in the country. And of course, the use of molecular biology, like for example, in detection of our aquaculture stocks. So like for example, our work in detection of different Anguilla species. As we all know that different species has its own price, and most of our works here in the Philippines are more on DNA barcoding in terms of application for policy, for development of policy, like for example in the work of Dr. Muji in Lidong and other wild stocks. So the application of this for our policy recommendations to delineate the stocks. And recently, there are works on the molecular markers in order to determine some traits of different species. Like, for example, in our tilapia program, the identification of salt tolerance tilapia and hold, and for improve freshwater stocks of our fish. And of course, for cryopreservation. So this photo is the work or from the Bureau of Fisheries and Aquatic Resources, or BFAR NFTC in Muñoz, where we have our gene bank for our tilapia. And recently, we are developing protocols in order to improve cryopreservation and to have this applied for our technology outer station in order for us to have easy access of different stocks in the region. So I believe we have only one in the Philippines, and it is located at the BFAR NFTC in Muñoz, Nueva Ecija. So yeah, just to show with you some works in salmon before that they have some researchers produced gigantic or giants salmon using biotechnology. So it's like an extraordinary salmon growth. I think it was around 1990s, and our some work on the transgenic in tilapia and in loach by other group of researchers. And in the Philippines, actually, we have developed our YY, and we have our improved Excel tilapia, and these are all made for traditional breeding and different hybrids. And now BFAR NFTC is also developing red tilapia for improved red tilapia for use in different environments, like in brackish water and in fresh water. So there are other applications of biotech, especially in aquaculture. For example, in our lab, in fisheries biotech. So we are on the developing kits or diagnostic protocols for the emerging aquatic diseases, like for example, viruses like TILV and streptococcosis in tilapia, and iridovirus in some of the commercial fish, for example, in milkfish and in grouper. And of course, development of molecular protocols for in applications in genetic research. And recently, we are working on assisted reproductive biotechnology, or ART in fish, reproductive biotech, using induced breeding hormones, which will be I'll be presenting later, and some of the natural product development in the lab using hormones, and our initiatives in reviving the indigenous species in the Philippines, like for example catfish, and in climbing perch. And of course, now we are into vaccines with our network in different countries. Like for fisheries, we are into collaboration with our counterpart in Taiwan. And I'll just show some of our work in how we develop some of our protocols in induced breeding of fish. So here I will be highlighting in the use of hormones in induced breeding of mudfish and catfish. And why we use this? Because it can regulate environmental condition or by introduction of hormones to stimulate factors that trigger reproduction. And using induced breeding hormones, it can promote ripening of gonads, and it also you can also control the release of sperms and eggs anytime if you wish to do some induced breeding protocols or apply breeding protocols in your stocks. So for induced breeding, it also to have an ease in fishing pressure, because as we all know that there are already declining in stocks, and it can aid species conservation for proper management. And of course, it's a potential for aquaculture. And increased productivity, so anytime you could have it. And you can provide fingerlings to our fisher folks, and it's also an alternative source of livelihood, and of course to increase income to our fisher folks. And recently, our work on induced breeding, like for example in mudfish, so we have optimized the protocol, and we have already our distribution of fingerlings to our fish farmers in order for them to stock for aquaculture purposes. So in order for them to have income out of this researches and innovations that we have for induced breeding. So in the Philippines, we are having production in different sites and optimizing and having trainings for this purpose in our fish farmers. And why is it essential to have the induced breeding in aquaculture? Because the technique is simple, and we does not need too much technical assistance or complicated protocols. And we can have our applied it to our breeder anytime. And we have targeted schedules. So as I've said, we can have our calendar our activities in order for us to, like anytime we could have the induced spawning, and we can have a pure spawn of fish under cultivation, and offers more controlled hatchery or aquaculture operations. So that's one of the applications of our induced breeding technique for production in order to increase for aquaculture production. And why is it that we need to have, or the mechanism of induced spawning in the process in fish? Because here we can alter different environmental factors that triggers reproduction. No? So for fish, actually, for example, the fish is already ripened or gravid year-round, but it needs some factors to trigger spawning. So here we can, using this technology, we can control and produce egg anytime if we get employed using these techniques. And the science behind this is to bypass natural biological process to speed up or to induce spawning in fish. Like for example, for some of the hormones that trigger reproductions. And recently, there are already commercial hormones. So this does not mean that the promotion of the hormones, but some of the works that being used in the lab for inducing hormones of the fish. And now we are working on the production of this hormone using organic hormones, like PG or pituitary glands of the fish. and in order for us to manipulate and produce fish anytime. And I believe there are some takeaways, and I think this is my last slides. So biotechnology offers the aquaculture industry a powerful set of tools to meet the increasing demand for seafood sustainability and food security. So recently, there are ongoing researches and innovations that drive progress and offer solutions to complex challenges, like, for example, the disease management, resilient to different environment, and resource optimization. So I believe that we are on the infant stage. However, we are on the competitive development for the protocols, especially in terms of molecular tools by its application in fish. So we also want to extend partnerships and collaborations in order for us to have a very good outputs and sharing of resources and researches in order for us to achieve a bit better food for our people, and for, of course, for security as a whole. So there are some references for the photos and other publications for this paper. So I think that will be all, and you can find us in BFAR, in FFTC, in the Central Luzon State University, San City of Muñoz, Nueva Ecija. Thank you very much.
Thank you very much, Dr. Cass. A virtual round of applause for Dr. Cass for his very informative presentation. May we put you on standby, sir, for the open forum later, which will be held following the final presentation. So again, reminder everybody, don't forget to send your questions via the Zoom chat box. Again, thank you very much, Dr. Cass. Okay, so moving forward, our final speaker for today acquired his bachelor's and master's degree on veterinary studies at Central Luzon State University. He graduated with a degree of Doctor of Philosophy, Infectious Diseases, Molecular Immunology and Microbiology in Hokkaido University, Japan. He was also a Senior Research Fellow of the DA Biotechnology Program. He formerly served as Chief of the Livestock Biotechnology Center. He is the chair of the Technical Working Group for the formulation of the proposed Joint Department Circular, which provides the framework for the regulation of genetically modified animals and animal products. And he is currently the director of the DA Biotech Program Office. He is also a career scientist. He advocates biotechnology as an alternative tool to increase livestock production in the Philippines, to improve food security and sustainability. His research interests include veterinary immunology, epidemiology, virology, molecular biology and diagnostics, vaccine and drug development. Let us give a virtual round of applause to Dr. Claro and Mingala. Good morning, sir.
Good morning to everyone. Thank you very much, Ms. Yen. Thank you very much for the participants. I will be sharing my slide to you. Just a moment.
Okay, so while we are waiting for Dr. Mingala to share his... Okay. I think your slide's already on the screen, sir.
Naka-slideshow na ba? Is it already on a slideshow?
Not yet, sir.
Not yet. Why is that so?
Okay, so while we are waiting, may we encourage everyone to send us your questions via the chat box. We have already received a number of questions, which we will discuss later following the presentation of Dr. Mingala. Sir, please take it away.
Is it okay now?
Yes, po.
Okay. So I will going to present to you the status of the regulatory guidelines for animal genetically modified animals in the Philippines. And it's, actually this was conceptualized, I think, around 4 to 5 years ago. And still, this is being assessed as well, and still undergoing some risk impact assessment or evaluation, so that it would be very fit to be used for the regulatory system for genetically modified animals in the Philippines. So the Philippines is considered to be the first ASEAN country to initiate a biotechnology regulatory system. In 1987, scientists from the University of the Philippines, Los Baños, the International Rice Research Institute, the Bureau of Plant Industry, and the PICARD of the OST formed a committee and formulated biosafety protocols and guidelines for genetic engineering and related research activities for the university and the IRI. The draft on Philippine biosafety policy was submitted to the Office of the President in Malacanang, and it was signed in October in the year 1990. As Executive Order 430, a series of 1990. The said executive order created the National Committee on Biosafety of the Philippines, or what we call now the NCBP. It is composed of representatives from the Department of Agriculture, the Department of Environment and Natural Resources, the DOH, or the Department of Health, the DOST, or the Department of Science and Technology, and some experts and scientists, and of course, members of the community. So the executive order was then followed by the issuance of the Department of Agriculture Administrative Order Number 8, series of 2002, as implementing guidelines for the importation and release into the environment of plants and plant products derived from the use of modern biotechnology. So in 2006, then President Gloria Macapagal Arroyo issued Executive Order 514 for the establishment of the National Biosafety Framework, prescribing guidelines for its implementation, reorganization of the NCBP. So this NCBP, or the DNR for particular, for evaluation of the environmental risk and impact related articles, and the DILG, of course, for the local governments, the DA for the agriculture, and then of course the DOS for health, and the DOST for the research. No? The DAAO8 was then replaced with the Joint Department Circular signed by the DA, DOST, DNR, DOH, and DILG, which we now call the Joint Department Circular. the DOST is tasked to regulate, as I said earlier, regulate articles, applications for contained use, and of course for research, and confined tests as well. No? The DA for the application for the field trial. So if the particular product is already transferred for field trial, the Department of Agriculture will take over. And it's also for the commercial propagation and transboundary movement of regulated articles. DENR for evaluation of environmental risk and impact of regulated articles. And the DILG is involved in supervising public consultation during field trials. So if you will notice, or if you will be able to read some issues regarding the sectoral petitions against a particular biotech products like GM corn or BT talong or BT eggplant. This is actually one of the main tasks of the DILG in pacifying or controlling the sectoral movements against a particular product that are being field trialed. No? So in this particular GM animal regulatory policy, what I have presented to you a while ago is all about the commencement of regulatory policies for agricultural and as well as other products derived from the use of modern biotechnology. And mainly, it was established mainly for crops. No? That's why we are tasked to draft another guidelines for the GM animal regulatory policy. And this is the one that I am mentioning earlier about the comments of this particular conceptualization. No? I think I said earlier 4 to 5 years ago. And until now, although it's as mentioned by Dr. Villanueva earlier, it is now being assessed at the level of the Department of Agriculture, and it's already passed through several consultations, no? Stakeholder consultations. But of course, it's not that easy because we still saw another level of assessment, which is the risk impact assessment that will be going to be done, of course, by the team and in coordination with the Department of Agriculture. So through the initiative of the Bureau of Animal Industry Biotechnology team, the necessity to establish guidelines for GM animals and animal products was recognized. Likewise, with the JDC for the plants and plant products, the proposed guidelines for animals and animal products involve the 5 other departments of the bureaucracy. And we are hoping that this particular regulatory policy will be assigned probably next year. So you will notice here in this slide, it's a series of 2022, but it's actually 2023 already now. So we are hoping that this will be signed by next year. So it will become series of 2024. This particular GM animal regulatory policy, the proposed guidelines is applicable to the following. No? Of course, earlier, Dr. Villanueva presented about animal or livestock biotechnology, and then Dr. Sureska presented about the fisheries biotechnology and the efforts through research and applications. This particular regulatory policy will cover both the 2 areas, no? The livestock and animal biotech and the fisheries biotech for genetically modified species. So as I mentioned, The proposed guidelines is applicable to the following: genetically modified fisheries and other aquatic resources, domesticated animals as well, and biological products used for animal husbandry and veterinary medicine. And the purposes of biological agents used for biocontrol derive from the use of modern biotechnology and containing novel combination of combinations of genetic materials. It is also emphasized in the proposed guidelines that the products of gene editing that do not contain novel combinations of genetic materials are not covered by this circular. So this is very clear with regards to this particular regulatory policy. And with this, this will be the biosafety decision points. No? It says here that in making biosafety decisions, the concerned agencies will be guided by the principles under the National Biosafety Framework, namely standard of precautions, risk assessment, environmental and health risk assessment as well, socioeconomic, ethical, and cultural considerations, access to information, and transparency and public participation. So all of this, the standards of precaution means that the risk, that the lack of scientific certainty or consensus due to the insufficient relevant scientific knowledge shall not prevent the concerned agencies from making appropriate decisions. So who are these concerned agencies? I will be discussing to you that later. In such cases, the concerned departments or agencies are expected to take necessary actions to protect the public interest and welfare. So it's always a kind of similar objectives with the crops, no? We are discussing here about the GM animals, so it's similar. The structure or the framework is similar in terms of the crops regulatory policy or the plant and plant products, the GM plant and plant products. The risk assessment shall be mandatory and central in making the biosafety decisions, consistent with the existing guidelines such as the NCBP and the Cartagena Protocol. Moreover, the concerned agencies shall also consider the environmental and health risk. The socio-economic, ethical, and cultural considerations shall also be taken into account, especially on the aspect of conservation and sustainable use of biological diversity. This is very important because we all know that the Philippines, as an archipelagic country, we have so many indigenous people groups, no? And we also want to conserve, no, the ethical and cultural considerations of these IPs. No? The right of the public and the stakeholders to information relevant to the biosafety decisions shall also be respected by the concerned government agencies and departments. So in this particular slide, based on the classification of regulated articles, the draft regulatory policy is categorized into four classifications based on their intended use. So there are four: research and the development of regulated articles under contained use. So this is actually much of the mandate of the DOST. And then we have the research and development of the regulated article for limited release into the environment. This is still under the DOST, but of course it will like complement with the DANR and DA as well. And then the commercial use of the regulated article with containment. So this will be, if this all about agriculture, and since this is this particular regulatory policy pushed by the Department of Agriculture, this will be the task now of the department. And then we have the commercial use of regulated article for general release. Based on these classifications, various agencies shall take part in evaluating and issuing biosafety permits. So for the research and development, the in-charge agency, as I mentioned earlier, is the DOST, specifically DOST Biosafety Committee. So there will be a biosafety committee within the various departments. They will be guided by the biosafety guidelines for the contained use of genetically modified organisms. For the limited release into the environment, still the DOST is in charge, as I mentioned earlier, in coordination with other concerned regulatory agencies, such as the Bureau of Animal Industry and the Bureau of Fisheries and Aquatic Resources. So since this particular regulatory policy will be covering both terrestrial and the marine or fisheries sector, the BAI and BFAR, which is under the Department of Agriculture, will be the leading agencies. No person or institution shall conduct R&D activities, including GM animals intended for limited release without the prior approval of the DOST Biosafety Committee. Limited release means small-scale experiment or field trials of a GM animal species performed under terms and conditions that mitigate impacts on the surrounding environment according to the Crop Life International. So I would like to emphasize here that because this is a regulatory policy for GM animals, we will be looking into the animal welfare, into the public health, and into the environment. So it's a kind of a One Health approach by the WHO and, of course, the World Organization for Animal Health. No? So for the commercial use, either BFAR, as I mentioned earlier, or BAI shall lead in assessment and issuance of biosafety permits. There are 8 uses identified. So you have here the 8 uses. For regulated articles under containment, we have the FFP, or the food, feed, or processing. And then we have the xenotransplantation, the bioreactor for medical pharmaceutical uses or biofarming. And then we have the bioreactor for industrial uses. Animal pets shall be regulated by BAI. Meanwhile, the FFP for aquatic species and ornamental aquatic species shall be regulated by BFAR. For the other possible uses, the lead agency shall either be BFAR or BAI, depending on animal species. So similar to what we have here now in the seminar, as presented by Dr. Villanueva and presented by Dr. Chiresca, that's also the what you call this, the division of the mandates for regulating a particular GM animal product. No? Either it's a product or it's a live animal. And now here, the policy on the commercial use of regulated article for general release, kanina contained use, ito general release into the environment, shall likewise be categorized into its intended use as well. This shall also be led by the BAI and the BFAR, depending on the animal species. The food, feed, processing animals, biocontrol for agriculture purposes, and animal disease control shall be, of course, led by the BAI. Meanwhile, the FFP for aquatic flora and fauna shall be led by BFAR. In this specific use, biocontrol for human health, household and industrial uses, the DOH shall lead in coordination with other concerned agencies. So there are, it's actually a kind of complicated scenario, but we will trying to smooth it by assigning the specific agencies, whether whether the Department of Agriculture or other departments in the bureaucracy like the DOH or the DOSP. So in this particular slide, the proposed guidelines shall follow this general procedure. First, the applicant shall submit its requirements to the BIFAR or the BAI, depending on the species of the product. The biotechnology office shall serve as frontline service and will determine the necessary actions for issuance of biosafety permits. The said office shall organize a joint assessment group. The application shall be forwarded to the joint assessment group form. The said group shall be chaired by the BIFAR and BAI or BAI and is composed of personnel from various concerned agencies based on the particular type of biosafety permit being applied as mentioned in the previous slides. The JAG or the Joint Assessment Group is in the middle of this slide, will make necessary technical recommendations on the application. The technical recommendation from the JAG shall be forwarded to the either BIFAR or BAI, and its director shall be by director and shall be a basis for granting or denying the application for biosafety permit. A regulated article may apply for the regulation. So before expiration of the biosafety permit, BIFAR or BAI may remove the regulated article from the coverage of this circular. If based on the nature of the regulated article will not pose greater risk to human and animal health, and of course to the environment, to the biodiversity that is conventional counterpart. So it's always there that we are focusing on the animal welfare, public health, and the environment. So the deregulation may be done in two ways. So this is the process. Number one is motu proprio, or on one's own initiative. And number two is through petition. Motu proprio, or in Latin for on one's own initiative, the BIFAR or BAI may consider the regulated article for its deregulation. The BIFAR or BAI shall declare such intention with supporting documents, of course, equivalent to the contents of a petition. If through petition, the applicant, among other requirements, shall state the factual ground why this circular should not apply to the regulated article, and published scientific literature relied upon by the petitioner. So there's always be a scientific proof that this particular article can be deregulated. No? It's not just like because it's already past the length of because it's already takes already years that is being regulated. But of course, if you want to deregulate it, you need to have a scientific proof to grant the deregulation. And I think this is my last slide. Yes, thank you very much. And I would like to emphasize that this particular regulatory policy is still with the Department of Agriculture and now being considered for the risk impact assessment that we hope could be performed as soon as possible so that it would be signed next year. Thank you very much for listening.
Thank you very much, Dr. Bong. At this point, may I now invite our other resource speakers, Dr. Marvin and Dr. Cass, to join us for the open forum. Okay, so we have received quite a number of questions So let me start with some of the questions were already answered by our guest speakers. So we will focus on those that have not yet been tackled. So the first one, it's from an anonymous attendee. Do we have a different decision tree on the regulation of GM animals? We have seen the decision on the regulation of plants and plants derived from PBIs yesterday. So I was wondering what is considered GM and non-GM in terms of animals. I think this question is for Dr. Bong.
I'm sorry, Miss Yen. I was able to get the gist of the question. Can you please repeat?
Okay, so do we have a different decision tree on the regulation of GM animals? We've seen the decision tree on the regulation of plants and plants derived from PBIs yesterday. So the question is about I think pertaining to plant breeding innovations.
Yeah, if I was able to get it right, I think it's the same, no? Because first of all, as I mentioned earlier, this particular policy is also based on the existing JDC1 for plants and plant products, GM plants and plant products. The only thing that is different from here is that there are two species or two commodities that are involved. First is the animal and the livestock and the fish. But of course, two different bureaus will also get involved in this. It's not like just the plant or plant products, which is the Bureau of Plant Industry. In here, Bureau of Animal Industry and the Bureau of Fisheries and Aquatic Resources will be the one leading this particular guide, using these particular guidelines. If there will be any applications later on. The effectiveness or the effectiveness of these particular guidelines will be assessed or evaluated later on when there will be an application already. Because as I said earlier, this is just a draft and is still being undergone risk impact assessment. So we hope that very soon this will be signed. And of course, similar to any other guidelines that we have, it can be possible for an amendment later on if it's not functioning well, or similar to the GM plant and plant products guidelines. They already amended this particular policy because of some issues on, I think, lengthy process. They streamlined it. So this GM animal will also be, probably in the future, would also be amended. But of course, let's wait first for the signing of this particular guidelines. So as I said, there will be no difference because this is also based on the GM plants and plant products JDC. Thank you.
Thank you very much, Dr. Bong. So may we move on to the next question from Mr. Josh Wilson. Can you speak of your marketing plan? Do you intend to label your innovative products on market and keep the environment safe? If it does have manipulation in use. I think this question would be applicable to our technology developers, so Dr. Marvin and Dr. Cass. Regarding marketing plan.
Okay, maybe I can answer first. Because majority of the animals, GE animals or GM animals that I presented were from researchers from abroad. Maybe for our side, because our group is mostly working on development of diagnostic kits for several diseases of concern here in the Philippines. So right now, the problem is more on the application of the patent. But I think once this application is okay and there will be some technology takers, I think there will be no problem. I cannot answer for those GM animals for production since as Dr. Mingala mentioned, the guidelines is still on the evaluation or risk assessment stage, and right now those technologies uh, that can be used here in the Philippines are still on hold, or actually even in other countries it's still not used for commercial use. They are just in the research facilities right now. So I think that's the only information that I can share. Thank you.
Thank you very much, Dr. Marvin. Dr. Cass,
Yeah. So good morning, and it's a part of our plan, the marketability plan, especially for the production of our GM animals. So, with regard to other countries, there have already, and so far in the Philippines, as Doc Marvin have said, we don't have. I mean, we don't produce yet GM animals, but in anticipation, it's part of our plan, and I think it's a must for food safety to have traceability of this GM animals. But I believe that for the GM salmon, they have already indicated some use, especially for that is in the frozen products for salmon. So thank you.
thank you very much, Dr. Cass. We have also a question about like GM animals which undergo regulated trials in GM animals. Is it the same process? I think this one was already answered by Dr. Mingala. We have the policy for limited release, which also pertains to regulatory trials. Moving on, there's another request or question. I'd also like to know about your Dr. Villanueva's insights on the challenges or barriers that our country is currently facing, as well as the opportunities for the advancement of animal biotechnology.
Okay. I think one of the biggest challenges that the introduction of animal biotechnology, introduction of this animal biotechnology here in the country is the acceptance. So if we don't have enough knowledge or information that we can share to the public, so that would be a big problem. So that is why this kind of activity, as well as the other activities that the Biotech Program Office as well as the three biotech centers are doing, is to do information dissemination to help increase awareness about the benefits of this agricultural biotechnology in addressing our problems. Aside from that, we also have other activities such as information dissemination to policymakers, so that they can help us in pushing the use of this biotechnology as a tool. I would like also to mention that aside from those information dissemination, it's also good that we should capacitate our partner stakeholders, be it private companies, SUCs, through trainings and workshops. Actually, right now we are having that kind of progressive manpower enhancement program to help capacitate our DA regulators as well as our partner researchers to do those biotech research that can later benefit our countrymen here in the Philippines. Thank you.
Thank you very much, Dr. Marvin. Okay, so moving forward, we have an FB live question from Mr. William Gamis. How do we prevent the unintended spread of modified genes to non-GM animal populations? What are the potential long-term effects of genetically modifying animals on biodiversity and ecosystems? I think I'll have to refer this question to Dr. Mingala.
Thank you very much, Michelle. Actually, it's a very challenging question, complicated question, because for now, all genetically modified animals, including the one that is reported or that are reported by Dr. Cass and Dr. Marvin, are being raised confined. So, for example, the salmon, the other animals that are sourced of bioreactors are all confined. So, if ever there will be some animals or livestock that will be raised or genetically modified, that will be raised, it will be confined. It's just that the issue for let's say issue on gene drive is another area for discussion. For example, if you will be like doing a genetically modified mosquito to prevent the spread of diseases for public health, how will you do that? So although there are already trials that have been made in Brazil or in that part of the world, Mexico, I think, using oxytec mosquitoes, there are also some sectors that are against to that because of the gene drive issue. In this particular GM animal policy, we remove the genetically modified mosquito. We didn't include that here because that is more on public health. That is not related to agriculture. So the Department of Science and Technology and the Department of Health will be organizing another TWG to look into that, because we cannot prevent that. If ever a genetically modified insect will enter our borders, no? So there will be a TWG. I don't know if they already started or they already have a special order on that. And also another is a possible genetically modified snails or even honeybee in the future, to increase their yields or to prevent the further spread of diseases that are transmitted by this particular animals. But that is related to agriculture. So we will be also answering those issues later on, because for now, all animals that are being raised, genetically modified animals that are being raised, are all confined. We don't have. There are no. I think no countries in the world that are completely release those animals into the environment yet. Thank you.
Thank you very much, Dr. Bong. So as I'm scrolling through the questions, I think most of the concerns are about risks of GM animals. And as part also of the Bureau of Animal Industry, I'd also like to clarify that the JDC is just a framework. So the details will, as mentioned by Dr. Mingala, will be indicated in the drafting of the implementing rules and regulations later on. So right now, what we have is the framework for the regulatory policy. So I think the next question is for Dr. Choresca. It is granted that biotechnology helps increase production in aquaculture In this regard, do you have data in terms of market demand, especially for red tilapia and market acceptability? If so, where can we possibly get it?
Thank you very much. So what a interesting question. So the B4, as I know, the B4 NFTC now is having research on the acceptability of red tilapia. And I know one PhD student, or she graduated already, one of her dissertation on the acceptability of red tilapia. So you may access the work of Dr. Mila Gross Apaga, Doc Mila, for that paper. And now we are promoting one of our researches in promoting red tilapia or broodstock development of red tilapia in brackish water pans. So part of that research of ours is on the market study, and we are now on the not developing, but we are on the market survey already, and hopefully we could have a very good results to share it with you. And I'm happy that if you are interested, we are happy to have more discussions with you in FBC regarding the promotion of red tilapia. And red tilapia is still the tilapia. It's the hybrids of crossing the different species. So I think it's more exciting. The taste is the same, and maybe there are some modification for different environment, but still it's tilapia. And I'm happy to have more discussions with you, especially for the acceptance and the marketability of red tilapia. Thank you.
Thank you very much, sir. So another question for you, sir. So we can still probably cater to a few more questions since it's already 11 a.m. So are there specific biotechnological solutions that can address challenges related to disease management and prevention in aquaponics, ultimately promoting fish growth and health?
I believe, of course, it's more on nutrition, because aquaponics, it's it's a complex environment and a very small environment. It's like a confined and controlled environment. So I think you must need, of course, the health of your fish, and you need to have the immuno or other nutritional requirements that your fish can stand with a controlled condition. And of course fertilizers, no, because you have to feed through using water solutions in your plants in order for you to have yields. So I believe that more on water enhancements, feed enhancements, especially feeding. So I think there are a lot of interactions. That's a very good model for biotech, because you are using a small space for your controlled systems. So I think there are lots of factors to be considered. More on maybe nutrition. If I were, so maybe you can feed a capsule in order for you that there's some, you can boost your fish to become bigger. And then for fertilizers for your plants, and water enhancer in order for you not to contaminate or to because there are lots of factors and to maintain the quality of the water.
Thank you very much. Thank you very much, Dr. Cass. We have another question from Dr. Gabby Romero. Will the BAI be able to meet the RIA requirement of ARTA? Thanks. So I guess the question is, shall be answered by the Bureau of Animal Industry. So, Sir Elijah.
Yes, hello. Magandang tanghali. So, bilisan natin para makakain na tayo. Yes, the RIA would be is ongoing. So as Dr. Bong said earlier, It will be in collaboration with the interagency technical working group. The RIA is ongoing right now. Right now, so we are drafting the PIA, the preliminary impact assessment, and as a result, yung sa PIS niya.
Hopefully, sir. Hopefully,
Oo,
if we can do it.
si Ma'am Yen ang ating hahanapin regarding dyan.
Uh,
Ang sisingilin natin.
Actually, we are seeking also the assistance and the expertise, of course, of our TWG members in the conduct of the RIA. But right now, we are still in the process of completing the PIA. So after the PIA, when the results will come in, and if it's determined to be a high impact, then we'll have to conduct the full-blown RIA. Based on the timeline, we are hopeful that we can complete it by early next year, if not this year. Thank you very much, Dr. Gabby. Okay, so we can still extend for a few more minutes to cater to other questions. Okay, so we've seen so much of the potentials of GM products for animal production, animal welfare, and food security. To level that off, what are the known, at least to this day, immediate and long-term risks of GM animals to the public health and the environment? So I think we would like to hear from each of our speakers, since this is pertaining to GM animals in general. So let's hear it first from Dr. Marvin.
Yeah, okay. So right now, if his question is in the Philippine side, as of the moment, we don't have the study yet because the guidelines are still on the draft. It's not yet reviewed, but maybe on the biotech crops, I know they are doing those assessment, since BT corn has been in the country for quite some time. But I think once this JDC for GM animals will be signed and the guidelines will be drafted soon, there will be a assessment that should be done to assess if what will be the effect of those GM animals that will be introduced here in the Philippines. Of course, the Philippine government would only focus on the what is best for the country. So as what Dr. Mingala said, if there will be some amendments that should be done later on, it's best to do that so that it would be for the welfare of the Filipino people. So I think that would be my comment on the question. Thank you.
Thank you, sir. Any comments from Dr. Mingala and Dr. Cas? Dr. Bong?
Yeah, thank you very much again to that question. Yes, it's actually very a concern, particularly there's a lot of what you call this fake news, fake news in terms of what we read in the internet, or even watch in the movies about genetically modified organisms or animals, etc. So sometimes it gives us scare, and a lot of issues regarding safety, food safety. But of course, this is the reason why we are having this particular GM animal regulatory policy, because we want to seriously assess the impact of these particular products in the animal welfare, public health, and environment issues. so far, since we don't have yet here in the Philippines, so far I think in other countries, we don't know any reports yet about the effect or adverse effect of animal products derived from gene modification to the human health or to the animal welfare or to the environment. We are just posing some questions because of the possible spread of a particular genetically modified animals in the environment that would affect the biodiversity. But for the health issues or animal welfare issues, we don't know any report at this moment with regards to the experiences of other countries that has already GM animals or animal products. Thank you.
Thank you very much, Dr. Bong. So one question also is related to that. So are there any long-term studies assessing the safety of biotech products, and what do they reveal about any potential health concerns? So we'll move on to the next question. Okay, so I think this one is pertaining about biodiversity. Will it not pose a threat if ever the existing species will be overrun by new breed of species, which is a product of biotech? Dr. Bong?
Sorry, will it not pose a threat? Existing species will be overrun by a new breed of species, product of biotech. As I have mentioned before, but your question is a very general question, not only as a GM animal. Are you also referring to any other breeds or that was as we discussed earlier? Biotech is not GM alone. No, biotechnology is a kind of tool to enhance the productivity of our livestock or fishery species. So we have the traditional, the modern biotech. So the one that we are doing traditionally, like for example, crossing the Brahman with the concentration or developing a lot of breeds in the swine industry, that's our biotech. And I think there are no adverse effect with regards to overrun the breeds. Is that a concern? This is just an additional kind of breed that would give a potential or increased productivity in terms of prolificacy in swine or milk yield in dairy animals. So I don't think there are other adverse effects. For example, probably a good example is the native animals that we have now. So is there a risk that our native animals will be overrun by hybrids? I think there is a possibility, but if we will be able to like create a market that are for the native animals. For example, a lechon that is more delicious. No, if we use the native animal, that's a market. So the production of a native animal will continuously prosper because there is a niche market. So I think that are some of the reasons that we need to be in our minds, so that we won't be like thinking always that there will be an adverse effect, not only to the health or to the environment, but also losing our breeds in the future.
Thank you very much, Dr. Bong. Dr. Marvin, do you want to comment on that?
Maybe no more. I don't have any comments on that.
Okay. Thank you very
Dr.
much,
Mingala
sir.
already explained it. Yeah.
Okay, so let's proceed to another question for Dr. Cass. What research has been conducted to assess the long-term effects and safety of consuming sex reverse tilapia, particularly regarding human health and development?
I believe that I think we have already some papers, but I forgot on the withdrawal of hormones in and its application to sex reverse in tilapia. So maybe if I could find that paper and if you could identify, I can send it to you. And of course, Dr. Rafi Guerrero is happy to answer your question because that project or that research has been, his work during the introduction of the sex reversal in tilapia in the Philippines. And I believe now that CLSU is having or developing organic testosterone for the application of from pine pollens. I mean, I don't know the exact product that they have, but that's also for the sex reversal of tilapia. And it's not a synthetic hormone, and it's an organic hormone. Thank you.
Okay, thank you very much, Dr. Cass. Okay, so in as much as we want to cater to all questions, we are limited by time. So once again, thank you very much to our resource speakers, Dr. Mingala, Dr. Villanueva, and Dr. Churesca. So a virtual round of applause to our guest speakers. Virtual round of applause po. Okay, so before we conclude the webinar, may we hear a few closing remarks or parting words from our speakers. Let's start probably from Dr. Marvin.
Okay, thank you, Ms. Yen. So, most of the GM animal products that I presented were from other countries, and it's good that in the problems faced by our country, the Philippines, it is good that it should be solved by our own researchers or experts or countrymen. So I hope we can reach more young and talented researchers, Filipino researchers who can work with us in developing biotech products that can address priority problems here in the Philippines. We hope that this JDC for GM animals will be approved soon, so that later on, if we can start right now to do some meaningful researches using modern biotechnology tools as an important tool, we can put it in the pipeline so that we can later on use it to address those problems in the country. So we are just offering our expertise and equipment and facility that we have, so that we can do more, we can work together and do biotech research. Thank you very much.
Thank you very much, Dr. Marvin. Dr. Cass?
Yeah, I believe that we are all welcoming to this new tools and techniques. We are already in the era of modern age, and now it's very competitive. So I'm also encouraging young scientists or our researchers and the likes, scientists and researchers, to work hand in hand to develop more for our people, So we will not compromise our, our constituents. We are developing products out of biotech to increase food for food security, and to provide our people. And one way of that is innovations using our tools in biotech. And I encourage everyone to to participate, and to be more proactive in biotechnology, especially for fisheries, no? For fisheries and aquaculture. And there are a lot more of tools to be employed or to be used to increase our food production. And there are a lot of ways to kill a cat. To develop the technologies, and it's a high time to do this. And using our guidelines to guide us, we can be more productive on research, especially for developing new technologies. And parting words, I believe that we are now living in an era of innovations, and research is very important to feed our people. So that's all. Thank you.
Thank you very much, Dr. Cass. And last but not the least, Dr. Mingala.
Yeah, again and again, I always impart this particular message to you by accepting that biotechnology is just another tool for agricultural increasing agricultural productivity. This is not the only tool, no. This is a promising alternative way in increasing agricultural productivity. And at the same time, we are developing our regulatory policy not because we just want to have another level or another concern that we always put, but because we want to really protect not because biotechnology is really very dangerous, no. We just want to be assured that a particular product, especially, would not affect the public health, the animal welfare, and the environment. So let's be very let's always bear in mind that all the efforts that we are exerting or putting in this particular area are not just because we want to do that. It's just because we have a noble objective to really, while increasing the agricultural productivity, we also want to protect our public, our consumers. Thank you very much.
Thank you very much, Dr. Bong. So before we officially end, I'd like to deliver a brief synthesis or summary of the discussions today. So from Dr. Marvin on his topic, genetically modified animals, developments in research and policy framework. It was highlighted that global food challenge is to produce more with less resources, and that to ensure enabling environment for biotechnology, there should be a clear, predictable, science-based and risk proportionate regulations, which is adaptive and responsive for uptake of new technologies. And there should be enough support to research and innovation, and of course, education and public awareness on the safety of these technologies. On biotechnology and aquaculture, meeting growing demand for seafood sustainably, it was highlighted that PH ranked 11th in aquaculture production in 2019, and that aquaculture is the country's main source of fish. However, there was recorded decrease in aquaculture fishes over the years due to large exploitation. And of course, interventions on aquaculture, specifically involving biotech, is on improved growth rate and productivity, increased resistance to pathogens, improved brood stock quality and control reproduction, and produce new or better products. And of course, specifically the project or the initiatives of the DA Fisheries Biotechnology Center is the induced spawning technology, which is aiming to ease fishing pressure and also to increase productivity and ultimately alternative source of livelihood for our farmers. And to conclude, it is important to take note that the draft JDC, as guided by the National Biosafety Framework, ensures science-based, transparent, balanced, and inclusive regulatory approach, espousing the following principles: the standard of precaution, risk assessment, environmental and health risk assessment, socio-economic, ethical, and even cultural considerations. Once again, thank you, everyone. And please don't forget to join us tomorrow for the third installment of the webinar series on biotech innovations for sustainable agriculture. Please remember that you will be getting your certificate after you have submitted the post-webinar survey, which will also be provided tomorrow. Once again, thank you very much from our partners, the DA Biotech Program Office, ISA Incorporated, Bureau of Animal Industry, the Biotech Coalition of the Philippines, and Corteva AgriScience. Thank you very much, and we apologize for the extension of 25 minutes, and we hope you will be having a great lunch. Again, thank you very much, everyone, and see you tomorrow. Thank you.
Thank you.
Thank you very much to our resource speakers and participants.
Thank you, all the speakers and participants, and of course, our partners. So you will be using the same link, okay? The same time. We have 2 foreign speakers and 1 local. So it's a very interesting session tomorrow on socioeconomics and social licensing. Bye.
Bye, ma'am. Thank you,
Bye.
everyone.
Thank
Thank you,
you.
everyone. Nice session.
Bye. Thank you very much.
See you again tomorrow.
See ya.
Bye-bye.
Bye.